Hey guys, this is Yuvraj and I'll be going over the course of AP Bio since the AP exams are now approaching and it's time for a quick revision. So we'll be going unit by unit over the course and today we'll be starting with Chemistry of Life, which is the first unit. So by now you should already know that there are three types of subatomic particles, which are neutrons, protons, which are found inside nucleus, and electrons that orbit the nucleus. Electrons are negatively charged and protons are positively charged, whereas nucleus are just neutral. Going on to the next important part is water. Water is a special molecule. It is one of the most versatile molecules and is required for life to exist. Why? This is because of water's polarity. Water has a slight negative charge near the oxygen and slight positive charge on both the hydrogens. This is because oxygen is more electronegative than hydrogen and it tries to snatch the electron from the hydrogens, and this makes the oxygen slightly negative. This gives water some interesting properties, which include water's high heat capacity, which basically means that it needs a lot of energy to change its temperature by 1 degree Celsius. And why is that important? It's important because if some natural calamity were to happen, all of and water's temperature were to change very rapidly, all the organisms in the ocean would die. And since life originated on oceans, on the big uh, water water biospheres, it would be really hard for life to reoccur. The second important part is water's expansion on heating, on freezing. Expansion on freezing. And this makes it so that water is less dense, is more dense than ice. And this is an interesting quality because water is the only molecule which solid form is less dense than its liquid form. And this is important. This is also important since, for example, we imagine a lake right over here. If water were to start freezing from the bottom up, it would freeze all the organisms as it freezes up. But since water freezes from the top up, and since ice is less dense, ice basically floats to the top and it basically gives the water this layer, whereas all down it's just water where organisms can photosynthesize, respire, do chemiosynthesis, etc. The third important point is that water has a high surface tension, and this basically allows insects to stand on water because water has the ability, water is basically cohesive. Cohesive means water is attracted to other type of molecules, like for example water can flow upwards a plant stem, and that is due to cohesive forces. Water is also adhesive, meaning it is attracted to itself. If you fill a water glass up to the brim, you will see if you keep adding more water, it will form a slight meniscus like this. It will overflow, but it will not spill downwards, and that is due to adhesive forces. The next important part of this unit is the four important biological macromolecules, which are carbohydrates, which I'll write carbs for short. Then we have proteins. Then we have lipids. And then we have nucleic acids. Carbohydrates are very simple molecules which have the basic formula, general formula of CH2O times N, meaning if C is 1, then H will be 2 and O will be 1. But if we take the example of glucose, in glucose carbon is 6, and if we multiply 2 by 6 we get 12, and we multiply O by 6 we get 6. So glucose is basically C6H12O6. Now the other important part of carbs is the disaccharides. Let me just erase this to make it a bit more clear. So disaccharides are basically two monosaccharides joined together by this bond, which is called a glycosidic bond. And when two monosaccharides combine to form a disaccharide, it is called a dehydration synthesis. And the reverse of this process is called hydrolysis. And polysaccharides are basically monosaccharides just put across from each other. Like this is one whole unit of a monosaccharide, then this, and then so on and so forth. Let me make it a bit more neater. So here, this will be a whole unit, then this, and this will keep repeating so many times. A very common example of monosaccharides is glucose, as I've already mentioned. Disaccharides examples are sucrose and lactose, which is found in milk. Many people are allergic to lactose, and that's because they don't produce an enzyme which is called lactase, which essentially breaks down the lactose. And examples of polysaccharides are starch, which is found in plants, cellulose, which is found on the tree barks, which is a form of cell wall, and chitin, which is found on insects' exoskeleton. Next, moving on to lipids, we have a glycerol molecule which is attached to 3 fatty acids chain, and this is a specific type of lipid called a triglyceride. You can memorize this by three fatty acids. Triglycerides are basically one glycerol molecule attached to 3 fatty acids, and the bond here is called an ester bond, the bond between the glycerol molecule and the fatty acid. Now one important point to note is that this molecule right here is an example of polyunsaturated fat, polyunsaturated, because it has multiple double bonds. And unsaturated means there's holes left where carbon can essentially fill in the gaps. You can see the double bonds by one bond and one bond on top of it. And if these bonds were to be replaced with hydrogen, these would essentially become trans fats. And a fat which has no double bond is called unsaturated fat. There's also different types of lipids along with triglycerides, which are steroids, which are hormones, and for the phospholipid bilayer, which is an important structure in cells, which regulates the inflow and outflow of a cell. And lastly, we have the triglycerides. And triglycerides are of 3 types. Let me just write that here. triglycerides, and these are of three types: polyunsaturated, actually four types. We have polyunsaturated, we have saturated, and we have unsaturated, and then we have trans fats. And one important thing to know about the lipids are they're essentially non-polar, meaning they they repel water. They are hydrophobic. They repel water because they're non-polar. They don't have any charge on any part of the molecule whatsoever. Whereas if we refresh ourselves, water has a charge on both the oxygen and two hydrogen molecules, which makes it polar. And one general rule to go by is polar molecules are attracted to polar molecules or ionic molecules, whereas non-polar molecules are just attracted to non-polar molecules. And lastly, we have the final biological macromolecule, which is the nucleic acids. Now there are two types of nucleic acids, which are the RNA and the DNA. Now some of the key differences: the sugar in RNA is ribose, as a sugar in deoxyribonucleic acid is deoxyribose, which is where it gets its name from. Then RNA is very primitive, which means it's belonged in the prokaryotes first, whereas DNA belongs in eukaryotes mostly. The bases in RNA are also different. RNA has a group which is uracil, and DNA has thymine. One important point to note that RNA does not have the base thymine, but DNA does, and DNA does not have the nitrogenous base uracil, but DNA does. Now, when we're talking about the structure of the nucleus, we have some important points. We have a deoxyribose. If it was ribose, it would have a O here, but it does not. So we have a deoxyribose sugar. So it has 5 carbons here, which will be 1, 2, 3, 4, and 5. And these are essentially called 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime carbons. Now, the structure is essentially attached to a phosphate group. The deoxyribose is attached to the phosphate group here. Let me make that a bit more clearer. It's attached to this phosphate group, and this part of the molecule is attached to a nitrogenous base. And these sequences of nitrogenous base is basically what is where the information is stored. So for example, here it could be guanine for me, whereas it could be adenine for you, or it could be cytosine for my mother, and so on and so forth. And one more important point to note, that is the DNA. There are two DNA strands, right? Since we call the DNA structure a double helix, which basically means it is a double helix like this. And while they revolve around each other, the directions are opposite. So for example, on this strand, the direction could be 3 prime to 5 prime going upwards, whereas here it has to be 3 prime to 5 prime, which means they run in opposite direction. This might not be clear, so I'll just make a linear representation. If this is 5 prime and 3 prime, the other has to be in the opposite direction. So this is the basically the whole structure of DNA, and it is also a type of monomer where the only thing changing is the nitrogenous base. And one more thing to note about DNA is negatively charged, which means if you run it through an anode or a gel, for example, this is a gel and this is the negative and this is the positive terminal, DNA will go this side. It will be attracted towards the positive side of the whole circuit. And the four types of DNA bases are adenine, which binds to thymine. The other is cytosine, which binds to guanine. They're paired with each other, and there can be no exceptions made here. Guanine, cytosine. Now we move on to RNA. RNA is basically the same as DNA, whereas one of the key differences being RNA is single-stranded. Means it runs like this. There's no complementary strand attached to it. One more difference is RNA basically has one oxygen attached here. If you remove this oxygen, this molecule becomes deoxyribose because an oxygen has been removed, and removed means deoxyribose. And say for example, if this were like this, if all the deoxyribose had the oxygen attached to it, and instead of thymine it was uracil, the molecule would essentially be an RNA molecule. And the pairing of the nitrogenous bases in RNA is basically adenine and uracil, and the second is same as DNA, which is cytosine and guanine. To sum it all up, we have 4 molecules. Oh, we're missing proteins. So now moving on to proteins, there are basically amino acids which have the general structure NH2, which binds to one carbon, which binds to a COOH group. Which binds to one hydrogen, and there's a side group here. And this is the basic structure of an amino acid, which actually is the building blocks of proteins. When these amino acids stack up, which means there's basically repeating units of these amino acids, the structure is called a polypeptide. Poly means many, so you should get poly means many, so you should know that there are many peptides there, and peptides is basically one amino acid. So for example, this is one amino acid, 2, 3, 4, 5, 6. This is a polypeptide chain, and the bonds in between is called a peptide bond, which holds the structure together. Peptide bond. One last important factor to know about proteins is that they have 4 types of structure. One is the primary, which basically tells us the order of the amino acids. So for example, if we name the amino acids on basis of the first letter, M, T, C, G, L, so on and so forth. One difference could be in one protein there's M, T, C, G, and L, whereas in another protein it could be M, C, L, G, T. This is the primary structure. Moving on to the secondary structure. We have to see how these bases interact with each other. So for example, in this chain, M could repel T and would form a bond with C, and T would attract G and repel L. So that would make it a form of beta pleated sheet. Secondary structures is basically divided into two structures. One is kind of like a paper folding many times. You have a paper, you fold it many times, and you have yourself a beta pleated sheet. The second is an alpha helix structure, which is basically like a rope and you twirl it many times, and this gives you the alpha helix structure. Then we have the tertiary structure, which is basically how these structures interact with each other. So basically there could be a helix, then a beta pleated sheet, then one more beta pleated sheet, and then an alpha helix structure. Whereas in another protein we could have a alpha helix, then another alpha helix, I'm really sorry for the drawing, another alpha helix, and then two beta pleated sheets. And these all structures are key in changing the function of the protein. So if you look here, this is basically more well done. So you can see helix here, this is basically the units, like how they're arranged. Next we have a alpha helix structure, then we see an beta pleated sheet, which then becomes an alpha helix, and then the quaternary structure. We have the fourth level of structure, which is the quaternary structure, which is basically groups multiple different polypeptide chains coming together. One we have like this, and then we have the other which is interacting with it here, and the other is making another structure here. So as we see here, this is one separate polypeptide chain, this is separate, and this is separate. And one more important fact to know about proteins is that proteins denature in high temperatures. And you might ask, what is denature? Denature is basically the changing of the structure, and if the structure of the protein changes, structure changes, the function also changes. Function changes, and that is detrimental for the protein taking effect. And to sum it all up, we have basically four types of biological macromolecules, and one of the most important molecule is water. And if you have noticed, in all these structures, the most common elements are nitrogen, which is found in proteins. We have phosphorus, which is found in nucleic acids and proteins, oxygen, hydrogen, carbon, and sulfur. And these are the building blocks of every molecule that makes up life. Thank you for listening for the summary of the Unit 1.